of chemical transformations that are catalyzed by enzymes. An individual chemical transformation is a reaction. And a plant cell typically carries out thousands of these reactions connected into a network. This is an image of a Arabidopsis metabolic network where the circles represent different metabolites belonging to different categories. Arabidopsis, by the way, is a model organism that a lot of plant biologists study, and it's very closely related to canola. So we're interested in understanding how perturbing a particular site in the metabolic network changes the network as a system, and why and how these changes occur. In order to be able to answer these questions, we need to have first a comprehensive view of the metabolic network, and we also need to be able to comprehensively profile the metabolites to understand what the changes are. So I'm going to tell you about a work that sort of addresses both of these points. First, how do we build a genome-scale metabolic network? In the last 15 years or so, there's been a lot of progress that's been made in the field, and many groups, including ours, have developed components that allow you to predict different components of the metabolism. And so in the last few years, we've actually combined these different components of the computational algorithms to build a pipeline that starts from a genome sequence and ends with a predictive metabolic network model. So the way it works is that we first take the proteins that are annotated from the genome sequences and predict metabolic enzymes using a set of curated, well-characterized enzymes as well as non-enzymes. And then using those predicted enzymes, we predict reactions and pathways from reference pathway databases. And we manually validate these predicted pathways using information in the literature. And then different tools are used to reconstruct predictive and quantitative metabolic network models. I don't have time to tell you all of the performance assessment that we've done, but I'll give you two metrics that show the performance. So our enzyme prediction pipeline, it's called E2P2, and it's indicated by these light blue bars, performs better than the existing algorithms on both precision, which is the false positive rate, and recall, which is the true positive rate. And when we compare our databases to other databases that have been developed by other groups on coverage, both the number of reactions and number of pathways are bigger in the databases that we generated. Our project is called PMN compared to other databases. So I think it's perhaps okay to say that what we've developed is safely what could be considered the state of the art today. And so using this pipeline, we've generated metabolic pathways, enzymes, and reactions and compounds for 17 plant species. And all of the data are available from our website called plantpsych.org. And you can, use the data in a lot of different ways. You can search, browse, download the whole data sets, and even put transcriptome and metabolome data onto the pathway context. And you can see that we predicted hundreds of pathways, tens of thousands of enzymes, and thousands of reactions and compounds. This is one image of a search that you can do for a pathway, and you will get all the information about the pathway, the compounds, the genes that encode the enzymes. These pathways are placed in the context of the previous pathway and the following pathway in the network. We provide information about the actual primary sources of where this data came from, whether it came from whether it has experimental support or it's just purely computationally predicted. And this is an image of a tool that's called Omics Viewer, where you can actually lay on to the metabolic pathways different types of omics data, like transcriptome and metabolome data. And then you can actually zoom in to individual pathways and get more information about particular reactions and get the sort of omics information in bar graphs or line graphs or heat maps, and then go to the sort of metabolic pathway context. These web tools are built by a collaborator, Peter Karp, at SR International. And so one of the first things that I asked for preparing this meeting is to ask, okay, what are the unexpected pathways that we found from these databases? So I define unexpected or latent pathways as those pathways that make metabolites that are known not to exist in the particular species. But then we predicted enzymes for the reactions, including the critical reactions. And when we looked at those hundreds of pathways that were predicted for these species, for many of the species, we did not find any unexpected pathways. The only exception was two. One pathway called strategy 2 iron acquisition. This is a pathway that exists in grasses where they actually produce compounds to chelate iron from the environment and then take the iron up through the roots. And it's not found in eudicots or non-grass monocots. But interestingly, we found the enzymes for all the reactions from a few dicots, including cassava and soybean. The other pathway we found that was unexpected was casbein pathway. Casbein is a diterpenoid compound that's an antifungal agent, and it's thought to occur only in castor beans. But we found the enzymes for the reaction that makes this compound in cabbage and poplar. So further work is required, of course, to see if these pathways really do exist in this unexpected species. But generally, we found that from surveying the entire sort of metabolic content that we could predict with the state-of-art technology, we didn't really find evidence of unexpected or potential latent pathways. But that could be because our sort of ability to predict the metabolic complements isn't really complete. And so one way of looking at how complete our sort of reaction repertoire is to ask how many of those reactions that we predicted are associated to metabolic pathways. So only about 75%, it's the majority though, of the reactions are associated with pathways, which means that we predicted about 25% of the reactions that are what we call orphan reactions. We predict enzymes for these reactions, but the reactions are not placed into any known metabolic pathways. Similarly, we predicted some reactions that belong to pathways that are known to exist in the organisms, but we couldn't find any enzymes for these reactions using the current technology. So certainly, our metabolic databases are not complete, and there's a lot more work to be done to identify these missing components. And I should also mention that most of these complements that we identified are predicted computationally. Okay, so a lot of work is needed to really experimentally define these activities. So now I'm going to switch gears a little bit and talk to you about some of the work we've done on doing metabolomics from single gene perturbations. So about 8 years ago or so, a few analytical chemists and plant biologists and bioinformaticians got together to form a consortium to actually generate a comprehensive metabolomics data set in Arabidopsis. So our group used about 136 knockout or overexpression lines in Arabidopsis enzymes that are either well characterized or not so well characterized, and using 11 analytical platforms, both targeted and non-targeted, profile metabolites from these mutants compared to wild type. And these plants were all grown in very controlled environmental conditions. So what we found was that we actually found close to 4,500 putative compounds from these profiles. And I think this is the most comprehensive set in terms of metabolomics in plants to date. Only about 30% of those putative compounds were chemically identified. And of the 1,300 or so chemically identified compounds, we were able to map onto a metabolic network. So from the compound perspective, our metabolic network is about 60% complete of what's been found, detected using metabolite profiling methods. So how good are these, before talking about how good the profiles are, I just want to say a little bit more about the genes that we profiled. So we actually selected both well-characterized genes that are predicted to encode enzymes, and the genes that are completely novel. And we chose these genes that are associated to all different domains of metabolism to increase the diversity of the genes that we profiled. And the profiles that we got were, I think, fairly high quality. And so when we look at the profiles of those genes that are well characterized, this is an example of a gene that encodes an enzyme that's involved in glycolipid biosynthesis in plants. Plants make glycolipids in two ways, one through the plastid and the other one through the ER and then plastid. The glycolipids that are made in the plastid actually make 18 and 16 carbon chain fatty acids. And so when we knocked out this gene that's involved in the plastid-derived glycolipid pathway, we would expect to see a reduction in the 18, 16 carbon fatty chain acid-based lipids. And then the flux would go through the ER pathway that makes lipids that have 18 carbon chain fatty acids. And that's exactly what we saw from the profiles. We saw a reduction in the 18-16 carbon chain lipids and an increase in the 18-18 carbon chain lipids. And so there are other examples of well-characterized genes that we saw expected metabolite profiles. And so when we surveyed this sort of diverse array of genes using these comprehensive profiling platforms, what are some of the general patterns that we observed? First, we found that these metabolic networks are really robust against perturbations. So most of the mutants we profiled only had less than 15 metabolites that were changed significantly compared to wild type. Over 80% of the mutants actually had less than 10 metabolites that were changed significantly. And this pattern is something we saw when we just looked at the well-characterized genes as well as when we just looked at the predicted enzyme genes. We also saw that those changed metabolites were specific to the mutants. So most of the metabolites that were altered only in 1 or 2 mutants. And so there was specificity also in the changed metabolites that suggests that the changed metabolites were sort of biologically specific to the genes that were altered. And then we asked whether those changed metabolites were clustered to each other in the metabolic network. This is an example of a gene when this red dot represents the site of perturbation and the blue dots represent the compounds that are altered in the mutant compared to wild type. And we saw that for many of the mutants that were perturbed, the compounds that were changed were clustered around the site of perturbation. Compared to when we randomly selected compounds from the network, they were well distributed throughout the network. So this indicates that the metabolite changes that we're seeing are likely due to the biological sort of effects of the genes that are perturbed. Now we're interested in using these patterns to actually go deeper into understanding the mechanism of change of these metabolites in the network using flux models. So to summarize, I've told you that much progress has been made in predicting metabolic networks in plants, and I think some of these tools could be used and potentially be useful in building hypotheses to assess both the intended and unintended effects or changes from genetic modifications. I've also told you that metabolic network representation, well, this is what I just said exactly. And I've also told you that metabolomics in the context of the metabolic network can help evaluate both the intended, I think, and unintended effects of gene modification more systematically and rationally. And finally, there are still a lot of missing components that need to be elucidated. With that, I'd like to thank the people that worked on the project. About half of my lab has worked on the project I told you about, and we're really grateful for lots of collaborators and funding sources. Thank you.